Physiology Pulse. Today's topic is synapse. So what is synapse? A synapse is where two nerve cells meet and talk. They don't actually touch each other, but they pass messages using electricity or chemicals. Okay? So the proper definition here you can remember is synapse is nothing but the junction between two neurons. It's not a physical joint. You can remember this. It's not a physical joint. It's only a functional connection. Right? In simple ways, you can remember where one neuron sends a signal to another neuron. Okay? That is synapse for y'all. What is the classification of synapse? So classification of synapse is again divided into two types, which is anatomical classification and functional classification. In anatomical classification, which is based on where the axon ends. It is based on the where the axon ends. Okay. So the type here is type 1 axonic, which means connection between axon and axon, right? And then think of it as what? Wire talking to another wire. It is nothing but a wire talking to another wire. Axon to axon connection, right? Next is axodendric type. In axodendric type, it is axon to dendrite, which is receiver wire. Okay, axon is the giver, then dendrite is the receiver wire. The connection between axon and dendrite. Okay, which is wire talking to a receiver wire. One wire to receiver wire. Axon is the giver, dendrite is a receiver wire, right? So that is why wire talking to a receiver wire. Next is axosomatic. Next is axosomatic type. What is axosomatic now? Axon to cell body. Axon to cell body. Okay. Wire talking to the main office. Now cell body is nothing but main office, right? So the wire which is axon talking to the main office, cell body. So now three types: axonic, then axodendritic, then axosomatic. So three types are here, which is anatomical classification. This is next. This is the image of the axonic synapse. Axon to axon. Okay, next is axodendritic synapse. Okay. This is axosomatic. Axon to dendrite, where wire talks to branches, okay, which is the most common type. It sends the signal to the receiver branches. This is the receiver branches, right? Receiver branches. Next is axosomatic. It is nothing but axon to the directly to the cell body. Directly to the cell body. Wire talks to the main office, which is very powerful. Okay. Directly it controls if the next neuron will fire or not. If the next neuron, this one, will fire or not, this will directly control. Okay. Think of it like dendrite is nothing but the branches that receive the messages. Soma, which is the cell body. This is soma, right? Soma is the cell body, main part of the neuron, whereas axon is the long wire which sends the messages. So axodendritic, wire to branches. This is wire to branches. This is branches, right? So wire to branches. Axosomatic is wire to the cell body, which is soma here. Okay, axonic is wire to the wire. This is how you can remember this. Next is functional classification. Now functional classification is based on how the signal is actually passed, how the signal passes, the function basically, how the signal passes. So it is again divided into two types, which is electrical and chemical synapses. Okay, electrical synapse is nothing but direct connection through gap junction. Okay, ions flow directly. Okay, so it is super fast and there is no delay. It is found in where? It is found in heart muscle, eye and brain. Think of it like a wired LAN cable. Instant transfer you can see here in electrical synapse. Whereas in chemical synapse, you can see it has a gap. In chemical synapse, it has a gap called a synaptic cleft. Okay. Now it uses chemicals, hence it is known as a chemical synapse, which is the neurotransmitters. So these neurotransmitters, they are used to send the messages. Chemical synapse use chemicals which are the neurotransmitters. So these neurotransmitters are used to send messages. Now they are slower, but can be excitatory or inhibitory. They are eventually slower compared to electrical, but it can be excitatory or inhibitory as well. So think of it like sending sending text messages. Okay, it takes a little bit of time. Okay, this is chemical synapse. Now this is the image of the electrical synapse and chemical synapse. Now you can see in electrical synapse, neurons are directly connected through gap junctions. This is the gap junction and it is directly connected through gap junction. Okay. So electricity flows straight from one to the next. So from one to the next, it flows straight. There is no gap in between. It's filled by the gap junction. So there is no gap in between. Okay. Instant transfer you can see because of this, because of the gap junction which is filling the gap. Okay. Now chemical synapse, there is no delay in electrical synapse, no delay. Whereas in chemical synapse, you can see little delay in passage of the messages because first neuron releases the chemicals. Here the chemical is released. Okay, first neuron releases the chemicals. Then they cross the gap. Okay, they cross the gap. This is the gap. Okay, they then start signal in next neuron. Okay. This is the gap. Okay. Then again it starts signal in next neuron. From here again it will start. So it takes a bit time for that. It takes a bit time. That is why we are saying there is delay here. Okay, this is presynaptic neuron, which is the sender. This is sender. Okay, then postsynaptic neuron, this is the receiver. Okay, gap junction, direct bridge, this is direct bridge. Bridge, okay, it is filling the gap. It is a direct bridge here. Whereas synaptic cleft here, this gap, it is a tiny gap here. This is a tiny gap. Only in chemical you can see this. Only in chemical you can see this gap. Because of this gap, there is a small delay. So electrical, fast but simple. Electrical is fast and simple. Whereas chemical is slow but more common. It is more common. It is more common and can control the message better. Okay, this is electrical and chemical synapse for you. Next is functional anatomy of chemical synapse, which is parts of chemical synapse. Okay. Here what can you see? Parts of chemical synapse. First we were talking about presynaptic neuron, right? This is presynaptic neuron. In presynaptic neuron, that is the sender. Presynaptic neuron, which is the sender here. It is axon terminal, which is end of the sender neuron. Okay. It has synaptic vessels as well. Presynaptic neuron consists of synaptic vessels. And what are these synaptic vessels? They are tiny sacs filled with neurotransmitter, which is the chemical. Okay. This is chemical synapse. So this is of chemical and which is used as the neurotransmitter. So it filled with neurotransmitter. Then has mitochondria as well, which is the powerhouse to make the chemicals. Mitochondria is also present, which is the powerhouse to make the chemicals. So that is presynaptic neuron. Next comes the synaptic cleft. Okay, synaptic cleft is tiny 20 to 40 nm gap between two neurons. Here, this is the tiny gap we are talking about. This gap. This is synaptic cleft, right? So this is a tiny gap between two neurons. Next comes the postsynaptic neuron, which is the receiver. Right? Postsynaptic membrane has receptors which locks the chemical keys, which are like the locks for the chemical keys. Okay. It also has cholinesterase enzyme to destroy the old chemicals. So cholinesterase enzyme will destroy the old chemicals. So in postsynaptic neuron, you can see all of these. You can see receptors which locks the chemical keys, and then also cholinesterase enzyme which destroys the old chemicals. Next is types of axon terminals. There are two types of axon terminals you can see. First is terminal knobs, which are swollen ends, which causes go signal. Whereas terminal coils, like the free ends, not knobs, the free ends, the wavy ends, they cause stop signals. Okay, they cause stop signals. Okay, next is the synapse. This is the synapse. Now can you see the neurofibrils? Mitochondria. This is the presynaptic neuron. Okay. This is the synaptic gap in which you can see the neurotransmitters. These are the synaptic vessels we were talking about. Chemicals, voltage gate calcium channels, mitochondria, powerhouse, neurofibrils. These are the ion channel receptors we were talking about in postsynaptic. They are receptors as well, right? So these are those receptors. This is postsynaptic neuron. This is the synaptic gap, and this is the presynaptic neuron. Okay, this is synapse. Next, the functions. Now you can see the functions of the synapse. Now what are the proper functions? First one, its main job is to pass signals from one neuron to another neuron, right? Pass signals from one neuron to another. Now again, there are of two types. Type 1, excitatory function. We are talking about type 1, excitatory function, whereas type 2 is the inhibitory function. Okay. So excitatory function is nothing but a go signal. Excitatory is nothing but a go signal. Okay. Now it makes next neuron fire with this function, creates the excitatory postsynaptic potential, EPSP. Okay, excitatory postsynaptic potential. Okay, now this EPSP, what will happen with this? How EPSP happens? Signal reaches the sender, calcium enters the sender. Okay, see here, calcium gated channels, right? Signal reaches the sender. This is the sender neuron. Then calcium via voltage gate calcium channel, the calcium will enter. Calcium enters the sender. After the calcium enters, what will happen? Vesicles release the acetylcholine in the gap. In this gap, acetylcholine is released. Oh God. Acetylcholine is released. Now acetylcholine will bind to the receiver. When it is bind to the receiver, sodium gated channels are opened here. Here, ion channels receptors are there, so sodium gated channels are also open here. Now sodium rushes in. Now after this, what will happen? Sodium will rush inside. Inside it becomes positive, where you can see mild depolarization inside. If strong enough, you can see the receiver fires its own signal. Now if that is, now if it is strong enough, if this is strong enough, this is a receiver, right? So this receiver neuron will fire itself. This will fire itself if at all it is strong enough. Now key here is EPSP, which is excitatory postsynaptic potential, is local, graded, and does not follow all or none law. It does not follow all or none law. Okay. Now next is the inhibitory function. Inhibitory function is nothing but the stop signal, stops next neuron from firing itself. Okay. It creates inhibitory postsynaptic potential. Okay. That is, this is the excitatory postsynaptic potential and this is inhibitory postsynaptic potential. Now how inhibitory postsynaptic potential happens here? It stops next neuron from firing. That is the function. Now inhibitory chemicals like GABA is released. It binds to receiver. It is released in the sender neuron and it binds to the receiver. Opens potassium and chloride channels here. Binds to receiver. After binding to the receiver, it will open potassium and chloride channels. After that, what happens? Potassium leaves or chloride enters. Anyone can happen. Potassium can leave or chloride can enter. Inside becomes more negative. After this, okay, after the opening of potassium and chloride channels, either the potassium leaves or the chloride enters. So what will happen with this function here? Inside it becomes more negative. When it is more negative, hyperpolarization is what we can see. Right? Now receiver becomes less likely to fire here. That is inhibitory function. Now inhibition types again. Types of inhibition. Type 1 is post-synaptic. We were talking about that, okay, last slide. Post-synaptic or direct, you can say. GABA acts directly on the next neuron. Directly GABA acts on the next neuron, causing it not to fire. Okay. Then presynaptic. In presynaptic type, it is indirectly acting. Okay. Axo-axonic synapse reduces chemical release from the sender. From the sender itself, the axo-axonic synapse, what will it cause? It reduces the chemical release from the sender to the receiver. When the chemical release is less, obviously the inhibition takes place, which is not directly acting on the next neuron, but indirectly it is affecting the next neuron. That is why it is presynaptic or indirect. Next is Renshaw cell inhibition. Now, Renshaw cell inhibition is nothing but a feedback loop. It is a feedback loop in a spinal cord to stop the overfiring which is happening here. Okay, that is the Renshaw. Oh, it is just feedback loop. It is in the spinal cord. It is stopping to overfire. Next is feed forward inhibition. Feed forward inhibition is in the cerebellum. It controls the movement of the precision. In feed forward inhibition in cerebellum, it is seen in cerebellum. It controls the movement of the precision here. Then reciprocal inhibition. When one muscle contracts, opposite muscle relax. That is reciprocal. Right? One muscle is contracting, opposite muscle is relaxing. That is reciprocal inhibition. Now, why this inhibition matters here? It stops too many signals at a time. Okay? Without it, you get continuously muscle spasms like strychnine poisoning or Parkinson's rigidity. In these conditions, you can see continuous muscle spasms, right? So, it will stop too many signals at a time. These inhibitions. So, inhibition matters a lot. Without it, you get continuous muscle spasms. Okay? Now, this is the image of a Renshaw cell inhibition. This is Renshaw cell inhibition. Okay. Now this is the feedback brake system for muscles basically. Okay. Now you can see parts here. Alpha motor neuron is one part here, which is the main neuron that tells your muscles to contract. It will tell muscles to contract, right? It will tell muscles to contract here. Alpha motor neuron, this one, what will this do? This wire going from the neuron to the muscle. This wire is going from neuron to muscle. This tells muscles to contract, alpha motor neuron, and this will go to muscle. This alpha motor nerve will go to the muscle and convey the message here. Okay, next is collateral. This is the collateral, right? This is a small side branch of that wire. This wire, alpha. So this is just a small side branch. Okay. Then this is the Renshaw cell. Small inhibitory neuron this is. Okay. This is a small inhibitory neuron. Okay, this is a small inhibitory neuron in the spinal cord. It is seen in spinal cord. So this is how the inhibition takes place. Okay. Alpha neuron will fire first here. Okay, alpha motor neuron will fire first here. This is where you can see the fire. After that, what will happen? It sends signals down to the nerve. From here, it will send signal down to the nerve. Okay. Muscle contracts. Collateral branch also activates the Renshaw cell. This is a collateral branch. This also activates the Renshaw cell. Okay. With a positive signal. Then Renshaw cell fires back again and sends inhibition. It will send inhibition to the same alpha motor neuron. Okay. Then it tells it to slow down or stop. Once it is done, it is sent. Then this will also send here collateral. Renshaw will again notice what is happening. Positive sign, this is happening. Then it will send inhibition to say that it is enough. You can slow down or stop the contraction. That is how you can see contraction and relaxation. Because the inhibition is taking place, immediately it will relax. Okay. It is something like a thermostat for muscles. Okay. When motor neuron works too hard, Renshaw cell will tell it to chill and not overdo it. It prevents muscles from contracting too much, keeps movement slow and smooth and controlled. Okay. Now this is the image of the presynaptic inhibition. This is presynaptic inhibition. Okay. Parts here you can see is the presynaptic inhibitory neuron. Okay, this is the bottom neuron. Sorry, this is the topmost neuron, presynaptic, top yellow neuron, this one. This is the blocker here for you. Next is presynaptic, which is the middle one, main sender. Next is post-synaptic, which is bottom neuron, main receiver. Okay. Now how it works? Normally Now, normally what will happen, presynaptic neuron would send signal from here. Presynaptic will send signal. Then postsynaptic neuron receives it. That is the normal. But here what will happen in presynaptic inhibition, what will happen, presynaptic inhibitor neuron, this one, presynaptic inhibitor neuron will first fire. Okay, this will first fire and it will give negative signal. It is the first one to fire. And it will give negative signal to the sender. This is the sender. So, it will give negative signal to the sender. That negative signal reduces how much chemical the sender can release. Okay. After which result you can see, even if sender fires, even if this one fires, even if the sender fires, even if it fires, the receiver gets a weaker signal. Even if it fires, what will happen here? It gets a weaker signal. Receiver will receive weaker signal. Even if it fires, what is happening? Because the first one is sending negative signals, this will have, again, it will reduce how much the chemical the sender can release. Because of this, it will reduce the chemical. After the reduction, even if the sender will fire, because it is releasing less of chemicals, the receiver will get a weaker signal. That is how this is happening. Think of it like a tuning down the volume before the speaker plays music itself. The speaker still tries to play, but the sound is very low. You're turning on the speaker is trying to play. Before it is still trying to play. Before that only, you thinking that it is going to be more, before that only you're reducing that sound. So hence the sound is very low. Okay. It filters unwanted. So what is the job of the presynaptic inhibition here? It filters the unwanted signals here. Makes you focus only on the important information. This is presynaptic signal, which is stopping the signal at the sender, not at the receiver. Here only it is stopping the signal. Here only it is happening. Sender side only you are stopping the signal. Okay, that is presynaptic inhibition. Next is properties. Properties of the synapse. You can see there are 5 properties here of the synapse. One-way conduction is the first property you can see, which is Bell-Mandy law. Bell-Mandy law. Signal goes only from presynaptic to postsynaptic here. It is never in the reverse side. It is from the presynaptic to postsynaptic, which is one-way conduction of the synapse, of the signal. Okay. Next is synaptic delay. This is second property. 0.3 to 0.5 milliseconds you can see here. Synaptic delay. Time taken to release the chemicals. Then cross the gap. Then bind to the receptor. You can see that, okay, in chemical side. Reason why reflexes aren't instant. You can see because of the release of the chemical, then it has to cross the synaptic gap, then it has to bind to the next neuron receptor. That is why you can see reflexes aren't that instant. It is used to check if a reflex uses one synapse or many. Synaptic delay. Okay. Next is fatigue. Now, if a synapse fires too much, chemical runs out. All the chemicals are running out because the synapse is firing too much. Then what will happen at some point of time? The signal will stop. So that is fatigue. Why you get muscles fatigue? Why will that happen? Because it protects brain from overexcitement. It protects the brain from overexcitement. Now the cause of this is the acetylcholine gets destroyed faster than it's made. Okay, compared to how fast it is made, it is getting destroyed faster, acetylcholine. That is why the chemicals are running out and the signal has to get stopped, so that the brain is protected from the overexcitement, that is fatigue. Now what is summation? Summation is nothing but small signals add up to make one big one. Summation is nothing but adding up. So small signals together, they are adding up to make one big signal. Now spatial summation and temporal summation. Two types. Spatial is many sender neurons fire at once. So many sender neurons fire at once, giving a big EPSP, excitatory this is. Okay. Whereas temporal summation is nothing but one sender fires again and again very quickly. This is many sender neurons firing at once. This is one sender neuron fires back to back quickly. That is big EPSP. Okay. That is summation for y'all. Next is electrical property, which is synapse shows, which is the synapse. This is the synapse shows go or stop, either go signal or stop signal. EPSP is nothing but go signal, excitatory, whereas inhibitory IPSP is the stop signal, that you remember, right? That is what the electrical property shows. So these are the 5 properties of the synapse. Now this is what we were talking about, spatial and the temporal summation. Spatial is nothing but the teamwork. It is like a teamwork. What happens here? Many neurons fire at the same time onto one neuron. So many are, simultaneously summation of many presynaptic terminals. So fire at the same time. At the same time they are firing, onto one neuron, one single neuron. But 5 pushing at once. Okay, it is like a person can't move, but move the car. Your car has a breakdown, a person can't move a car, then 5 people at a time they push a car at once so that the car can move. That is how it is working here. So these are the 3 friends who are trying to push this neuron so that it actually fires. So all these small signals add up, causing neuron reaches the threshold, fires an action potential, after which it fires an action potential. Now next comes the temporal summation. It is nothing but a rapid fire. Okay, what happens here? One neuron fires again and again very fast onto the other neuron. So this is the one neuron which is firing back to back, 1 2 3 4 fires. You can see repeated stimulation of one presynaptic terminal. Repeated stimulation, repeatedly it is firing. Okay, now think of it like hitting a nail with a hammer. Hitting a nail with a hammer, if you want the nail to get inside a wall, you are going to hit it back to back, right? One tap doesn't work. You have to give 5 to 6 quick taps so that the nail goes right inside the wall. So signals pile up quickly before they fade here, and the neuron reaches the threshold, causing it to the fire. So spatial is nothing but many places. This is many places, same time. This is one place, many times. Okay. Next is the convergence or divergence. After summation, you can see convergence or divergence. So here you can see many neurons to one neuron is convergence. Divergence is one neuron to many neurons. So here in convergence, you can see it helps the brain combine info from many sources. Whereas divergence, it helps spread one signal. It will spread one signal to many areas. This is from here. Spread to many areas. Okay. Now, super simple recap you can see here. Synapse is nothing but it is a chat point between two neurons. Electrical equals to the wired, which is fast. Chemical is wireless, which is it uses chemicals, neurotransmitters, and it is slower. Now EPSP, it is go signal by letting sodium in, whereas inhibitory IPSP is stop signal by letting potassium out or chloride in. Now key rules here you can remember is one-way traffic. If it is one-way traffic, small delay you can see, but it gets tired, small signals can add up. Now brain wiring is nothing but it uses the convergence to focus information. Like it helps brain to combine information from many sources, right? So it focused in the information into. Okay, next is the divergence to broadcast the information. To focus what the information is, it is coming from the convergence from many sources, right? What it is. Then to broadcast it, to spread the signal, you need divergence. That is what here we are saying about the brain wiring. Now this is how your brain controls everything from moving your finger to feeling emotions, all through the tiny chemical text at synapses. Okay. Now this is the convergence and divergence image. This is how it looks. This shows how neurons connect. Many to one, one to many. This is many to one, this is one to many, right? Now convergence, we'll be talking about convergence, which is the many to one. What happens here? Many presynaptic neuron. This is many here. And this is one here, right? So many presynaptic neurons send signals to one postsynaptic neuron. This is one here. This is many here. Okay. Now many presynaptic neuron will send signals to one postsynaptic neuron. Think like 5 people shouting information to one person. 5 people are giving information to that one postsynaptic neuron. It helps combine information, many pain sensors to one neuron, after which the brain feels ouch in one spot. Okay, that is what is happening. Next is divergence, one to many. With one to many, what happens here? One presynaptic neuron sends signals to many postsynaptic neurons, like one person sending a group text to 5 people. So it is like spreading information, right? So that is what it is doing. One motor neuron, many muscle fibers, then the whole muscle contracts. One motor neuron will send information to many muscle fibers so that the whole muscle contracts at one time. That is the information which is what the one motor neuron is passing to the muscle fibers. So this is synapse for y'all. Thank you for watching. Keep following.